We analyzed metal ion binding T4 lysozyme as an intramolecular protein purification tag compatible with X-ray crystallography. This was done by Bora, Bomlova, Chalupska, Danbankova, and Klima. It was published online at proteinscience.org on March 25th of 2017. The enterobacteriophage T4 synthesizes a lysozyme that hydrolyzes the linkages between acid and amino residues, which breaks down peptidoglycan. This is a mesh-like outer layer in the plasma membrane of most bacteria cell walls. T4 lysozyme is known to induce crystallization in proteins by replacing either the N-terminus or an intracellular loop of the target protein. It is a good protein tag due to its ability to maintain its folded shape despite amino acid residue substitutions. The authors created a metal-binding T4 lysozyme mutant known as MBT4L. This has been shown to be a superior protein tag for immobilized metal affinity chromatography, or IMAC. MBT4L allows for higher levels of purification and improved solubility of target proteins. Also, the authors found that MBT4L, due to its crystallization characteristics, can be used with protein crystallization to identify the structure of unknown proteins.
So how did they make this T4 lysozyme? Well, first they started with a truncated T4 lysozyme, which was designed by previous researchers. These researchers changed the lysozyme by removing the N-terminal lobe, which was known to be very flexible, and they replaced it with a short glycine-serine linker. What this did was it inactivated the enzyme, but it also improved crystallization, which were two things the researchers in this experiment desired. The researchers in this paper then went on mutating this lysozyme by adding 6 histidine point mutations using mutagenesis kit. The criteria for these histidine mutations was that they wanted them to be distal to the N and the C termini. They also wanted the histidines to be solvent accessible, and they wanted them to be sterically capable to form bonds with the chelated metals, which in this case are the nickel ions. The mutations were determined where to be placed using homology modeling and a protein database. So they looked at the best areas that these residues could be placed in this sequence without forming any, resulting in any big structural changes of the overall protein. So the places were chosen are shown here in red. So you can see the 6 different residues placed. They then expressed this metal-binding T4 lysozyme in E. coli. In order to test how these mutations had affected the overall structure of the lysozyme, they used protein crystallography, which was an X-ray, and compared it with the truncated T4 lysozyme. So that was their model. So when looking at that, they saw that there was a high-resolution structure for the metal-binding T4 lysozyme of 1.81 to 1.75 angstroms, which is right around the ideal of 1.5 angstroms. Also, it had a high completeness percentage of 99.45, and the Ramachandran plot gave a 99% value, showing that 99% of the bond angles were the same as those found in the truncated T4 lysozyme. Also, it resulted in a small average B factor of 36.4, which showed that there was a small amount of flexibility in this metal-binding T4 lysosome, which is what these researchers desired. Overall, it was very, very similar to the truncated T4 lysozyme. And actually, they also determined that it was superimposable on the wild-type T4 lysozyme. As you can see here, gray is the wild type, and the colored is the metal-binding mutant. And the only difference is this in the end lobe area, which is all gray, because that was cut off in the metal-binding mutant. So the researchers concluded that mutagenesis they had performed had no significant effect on the crystallization of this T4 lysozyme. They then wanted to test how this metal-binding T4 lysozyme would interact with the nickel ions in a crystallized structure. So again, they used protein crystallography, the X-ray, but this time they examined the anomalous scattering of the nickel ions. To do this, they used both single-wave anomalous dispersion and also molecular replacement single-wave anomalous dispersion. Both of these gave evidence that 2 metal-binding T4 lysozymes would bind with 4 nickel ions, as you can see in this image right here. So top and bottom are the metal binding T4 lysozymes. In the middle, here you can see the histidines attached to 4 nickel ions. So that's the crystal that it would form. They also wanted to test how this metal binding T4 lysozyme would work as a protein tag in IMAC, which is the mobilized metal affinity chromatography, and a general fractionation purification method for proteins. So first they needed a protein, so they used the known protein PI4K2A. The reason they used this one was that it was known to have a post-transitionally placed portion of it, which they could easily then replace with the T4 lysozyme. But they also needed a protein to compare the metal binding T4 lysozyme to. So they made a different recombinant protein, as you can see here, with the histidine 6 tag, which is a commonly used tag in IMAC. So they had that one, which they also added the truncated T4 lysozyme to, just to make sure that adding in that small lysozyme was not overall changing the protein structure in any way or making any big differences, to make these 2 recombinant proteins as similar as possible. So here you can see there's just this where they inserted the metal binding T4 lysozyme in the middle of the end lobe. They then expressed this in the E. coli, but they also wanted to compare affinity. So both these recombinant proteins were bound to the his trap HP column, and then they were eluted from this column with a gradient of imidazole. This elution they then characterized by mass spec and by SDS page for further analysis. So as you can see here, the yield overall for the metal binding T4 lysozyme in the E. coli was much higher, and it was at milligrams per liter, so it's much higher here. And then in the elution over here, you can see it for the metal binding, it peaks at around 10, which is a much higher concentration than in the one with the his tag, which peaks at around 6. So this shows that there was a higher affinity in the metal binding for the that tag. Also, here's the SDS page results, and you can see these are just protein bands here, and they just wanted to make sure they traveled to the same distance to show that there were no overall major changes that these different tags had on the protein.
To summarize, the first part of this research was to design and verify that the novel T4 lysozyme proteins could form crystallizable structures by binding to nickel ions. NBT4L was shown to effectively maintain its folded structure and composition of the wild-type lysozyme. The charged histidine mutations on the surface of the mutant gave the protein a polar nature, making it more soluble. In order to synthesize these intramolecular protein tags, they were generated using bacteria. The bacteria used was E. coli, which does contain N-terminal peptidases that can denature the N and C terminus of proteins. Therefore, the exact domain boundaries of the bacteria were no longer necessary to know, making it easier to synthesize the protein without producing a damaged tag. Further testing of the protein structure verified that it can form covalent bonds with nickel ions, and it was assumed that the full structure of the mutant protein could be achieved with MRSAD. This is important because if the structure is known, then when crystallography is performed on the target protein, the intramolecular tag can be ignored, therefore allowing researchers to deduce the structure of the unknown protein. When thinking about its use on a mass scale, NBT4L is more desirable than the more commonly used His6 tag because it has a greater affinity for the nickel ions and could be produced in higher quantities by E. coli bacteria. Also, the metal eluted from the column of the IMAC with a higher concentration. Therefore, since it has a higher affinity for the nickel ions, it is more effective as a purification agent for other target proteins comparable to the more commonly used His6 tag. Its one drawback as an intracellular protein is that the sequence of the target protein is unknown or difficult to predict and makes it difficult to tag that target. This is why it was desirable that the His6 attached to the end of the end lobe and compared to the NBT4L which attached to the middle of the end lobe, even though this quality protected it from degradation. Overall, the authors deduced that NBT4L lysozymes are shown to be new and better intramolecular protein purification tags for IMAC due to its ease of synthesis, greater affinity, and resistance to degradation.